Good afternoon and welcome everyone to FDA Grand Rounds, where we showcase FDA scientists on the great work that they do to advance regulatory science here at the agency. My name is Rakesh Raghuvanshi. I'm in the Office of the Chief Scientist, which is the sponsor for FDA Grand Rounds. Today's Grand Rounds is going to be focused on forensic proteomics applied to FDA-regulated products, and we're very grateful to have Dr. David Skelton of our National Forensic Chemistry Center with us. Dr. Skelton is a chemist at the National Forensic Chemistry Center, where he serves as an expert in the fields of forensic proteomics and biotherapeutic characterization. His focus has been on the development of proteomic techniques for the analysis of FDA-regulated products. Additionally, he's served as an instructor for multiple FDA training courses pertaining to mass spectrometry. So we're very fortunate to have you here with us, Dr. Skelton. Thank you so much for agreeing to speak, and the floor is yours.
Thank you for that introduction, and I'll start off by saying thank you for allowing me to present today on some of the proteomic work that we do here at the National Forensic Chemistry Center. So before I get into the presentation, I need to state that anything shown in this presentation does not necessarily represent the official position of the U.S. Food and Drug Administration. An outline for today's talk, we will go over a brief introduction to proteomics, discussing some of the techniques that we use here at the National Forensic Chemistry Center for these proteomic analyses and how we apply them to some of our samples. And then we will go over some specific cases that we have encountered recently here at the lab. So proteomics is concerned with the analysis of peptides and proteins, which are chains of amino acids. They are differentiated from each other based on how many amino acids are in that chain. So anything less than a 50 amino acid sequence is referred to as a peptide. Anything more than 50 amino acids is referred to as a protein. We routinely analyze both peptides and proteins here in the lab, as well as glycoproteins and monoclonal antibodies, which are special types of proteins with unique modifications on them, which we will discuss and show examples of later. And I put this on here. We also analyze glycans, which are biopolymers with sugar subunits, as well as oligonucleotides in the most recent years, namely mRNA. While neither of these are composed of amino acids, so they're not proteins or peptides, we do analyze them here in the lab, and they're listed here because they're analyzed using very similar techniques as the proteomic techniques that we'll be discussing today. So our samples will come to us in the lab based on in any number of matrices, including powders. We get in liquids, both for injectables, as well as oral suspensions, nasal sprays, eye drops. We also routinely analyze capsules and tablets, a multitude of food products, as well as various creams and cosmetics. When these samples arrive to us, we have any number of proteomic techniques that we can employ, depending on what information we are trying to ascertain. We have mass spectral techniques, mainly using liquid chromatography, mass spectrometry, or LCMS. We use these techniques to determine the unique amino acid sequence of the protein, as well as to identify any modifications to it. We also have electrophoresis techniques that use a polyacrylamide gel to separate and detect the proteins based on their molecular weight or some other physiochemical property. And we utilize immunoassay techniques such as Western blot and enzyme-linked immunosorbent assay, or ELISA, where the protein is detected based on its interaction with a targeted antibody. For the purpose of today's talk, I will only be focusing on the work that I am involved in, which is the mass spectral techniques, primarily using LCMS. So since proteins and peptides consist of amino acids linked together in a unique sequence, for mass spectrometry analysis is that unique sequence that we want to identify. The reason being is that the sequence of amino acids in the protein determine the structure of the protein, and it's the structure of the protein that determines its function. And any modification in that sequence can potentially alter the structure and therefore the function. So when we utilize mass spectrometry in our analyses, there is always at minimum two questions we want to answer. One, what's the unique sequence of amino acids in that protein? And two, what, if any, modifications are present? In proteomics, these modifications are called proteoforms. This would be analogous to the nomenclature isoforms that's used in small molecule analysis. So to understand how we can sequence these using mass spectrometry, we first need to understand how these individual amino acids are linked together in a protein. So there are 20 naturally occurring amino acids that occur in proteins. Each amino acid has the structure of a primary amine and a carboxylic acid connected via a central chiral carbon. And it is the side chain denoted by the letter R here on this slide that dictates or determines the identity of the amino acid. So each of the 20 amino acids has a unique side chain or R group coming off. And when these are linked together to form a protein, the carboxylic acid of one amino acid interacts with the primary amine of another amino acid to form an amide bond. And when we read the sequence of amino acids, we always read starting with the N-terminus amino acid, which is the amino acid with the primary amine, and we read to the C-terminus amino acid, or the one that has the carboxylic acid. So in this case, we had 3 amino acids that combine together to form a tripeptide. They are phenylalanine, aspartate, and alanine, and they are denoted here by their single letter abbreviations, FDA. So while I enjoy talking about mass spectrometry, I won't go into a great amount of detail on it today just for the sake of time. But as a general overview, what we do when we analyze these via LCMS is the peptides are injected into a liquid chromatography system. The peptides are separated from one another as they travel through a column. As these peptides sequentially elute from the chromatography system, they are transferred into the gas phase via the ionization source of the mass spectrometer. They are separated according to their mass to charge ratio in the mass analyzer, and then they are detected and a mass spectrum is generated. So in this example here, we have a peptide with a mass to charge ratio of 819.28002. And as we stated, it's the unique amino acid sequence that we want to ascertain from this information. So based on just the mass alone, if we assume this is composed of only the 20 possible amino acids and there are no modifications present, then even if we are utilizing a highly accurate mass spectrometer that has a mass error of no more than 5 parts per million, we have over 360,000 possible unique amino acid sequences that could be attributed to that mass. So the way we determine which of those 360,000 sequences is the correct one is by fragmenting the peptide, where in the mass spectrometer, we break the peptide down and we look at the fragment ions that come as it interacts with gas molecules in the mass analyzer, which cause it to fragment. The reason this is used in determining the sequence is because peptides will fragment in a very predictable way in the mass analyzer. They will fragment along the amide bond of all the adjacent amino acids. Each time one of these amide bonds is broken, 2 fragments are generated. The fragment that has the N-terminus amino acid, known as the B ions, and a fragment that contains the C-terminus amino acid, known as the Y ions. Each of these amide bonds are fragmented in the mass spectrometer, and based on this peptide sequence, we can predict what all of the B and Y ions would we see in the mass spectrometer, what their amino acid composition would be, and we can convert those into the theoretical masses that we should observe in the fragmentation mass spectrum if in fact our peptide does have this unique sequence. So knowing how they fragment, if we go back to our MS2 data, we can then try to ascertain the unique sequence here. So looking at the ion at 148, that corresponds to an N-terminal phenylalanine shown in blue here. If we look at the ion in red below it, that corresponds to a carboxylic or a cysteine with the carboxylic acid still on there. So that means it's our C terminus. And from here, what we start doing is we just start looking for mass differences that correspond to amino acids. So the difference between 148 and 263 corresponds to the amino acid aspartate, which is denoted by the letter D here. And we do this for all of our B and Y ions. We continue this process just looking for mass differences that correlate with the known 20 amino acids. And we keep going until we work our way back to the precursor mass. And so now we have here a candidate peptide with the sequence FDA in FCC. We can then annotate our experimental mass spectra with the B and Y ions. We compare this with what would be predicted based on the known fragmentation of the peptide. And we see that they match up, and we are able to narrow down our possibilities from 360 something thousand to one unambiguous sequence of FDA and FCC. So I made the example nice and pretty, but let's see how it actually works when we're dealing with a sample that we get in the lab. When we obtained this sample, it was an unlabeled vial containing a white powder. It was suspected to be a GLP-1 agonist. When we ran it on our LC-MS system, we saw only one peak eluding from the chromatography, indicating we only had one peptide present, and the corresponding mass spectra obtained. Each of the ions listed here are the same peptide, just at different charges. So the 1029 is the peptide with a charge of 4, denoted by the letter Z there. And the 1372, it's the same peptide, only now with a charge of 3 instead of 4. So we compare these masses with an in-house database we have of all the known biotherapeutics, both proteins and peptides. And we see that this mass corresponds to semaglutide. But as we saw earlier, the mass doesn't tell us anything about the sequence, and ultimately it's the sequence that we want to know, because that's what determines the identity. So we fragment our peptide ion, and we generate this fragmentation mass spectrum here. We follow the same procedure that we did before, where we are looking to assign the B and Y ions to annotate our experimental spectra, and we compare that with the predicted B and Y ions we should get from the known peptide sequence, which has this amino acid sequence here. For semaglutide, there are 2 amino acids that I've highlighted here in red that are of interest to us. The first being the AIB, which corresponds to alpha amino isobutyric acid. It is not one of the 20 naturally occurring amino acids that are observed in peptides, but it can be synthetically added in the laboratory. It is known that semaglutide has this in the 2nd position, and we do observe that here. Also, the lysine, which is denoted by the red K there toward the middle of the sequence. Semaglutide is known to have this structure as a modification on the side chain of that amino acid. So when we compare the predicted B and Y ions we should get from cleaving each one of the amide bonds between each of the adjacent amino acids, compared with the mass differences that we see in our experimental data, we actually can see that we can account for all the B and Y ions observed. So we can unambiguously assign the amino acid sequence as being that to semaglutide. We can also localize in the position number 2 the synthetic amino acid modification. And we can say that this lysine has a modification that has a mass that corresponds to the structure. However, we have to look at what our data is showing us here, because what we're looking for are mass differences that correspond to these different amino acids. And so for this lysine, what we've done here is we've said this lysine has a modification on it that has this mass that corresponds to its structure, but we haven't said anything about the structure of that mass. We just said it has a mass on there that has that corresponds to its structure. It could be a different structure with the exact same mass. So what we have to do is elucidate the structure of that side chain before we can say this is in fact semaglutide that's in here. And what we do is we go about this the same way as we would with the peptide. We predict the likely sites of fragmentation along the amide bonds in this side chain, and we start looking for the corresponding B and Y ions that would have those different fragments on them. So if we have a B ion or a Y ion that has this lysine in it, we would look to see, does that lysine have a modification that has this intact mass? And does that lysine also have a modification where we can see one of the masses that's resulting from each one of these fragmentation events? And what we can see in our data is we are able to account for all of these fragments of the side chain in our data. So this allows us not only to determine the amino acid sequence and to localize the sites of modification, be it a synthetic amino acid or one of the modifications on the side chain of the lysine, but we are also able to elucidate the structure of that synthetic side chain on lysine. This is compared with the certified reference standard of semaglutide, and we are able to confirm the presence of semaglutide in this unknown powder. So the next step that we take for these samples is we always do impurity profiling. We are looking for what else could be present in there. We are looking for byproducts from the synthesis process, as well as any adulterants or contaminants that we could observe. So in this case, we had a unknown powder that was suspected to be a GLP-1 agonist. Using the techniques that we previously discussed, we identified the GLP-1 agonist, and we saw multiple other lower abundant chromatographic peaks that we sought to determine the sequence of those impurities, and as well as localize any modifications that we observed. So here's our LC chromatogram, the base peak showing the GLP-1 that we were able to identify. And then if we zoom in on this region here, this is where we see these smaller abundant chromatographic peaks. Applying the same techniques that we have been using, we were able to identify oxidation products. So these are amino acids that have been oxidized at specific locations in the GLP-1 sequence. This DES 1 through 9 is proteomic nomenclature that indicates that the first 9 amino acids on the N-terminus side of the GLP-1 agonist were missing. So this could either be a result of an incomplete synthesis, or it could be a breakdown product. We observed synthesis byproducts. So we have an acetaldehyde shift base. We also observed formulation of the N-terminal amino acid on the GLP-1. These result from reagents used during the synthesis process that produce these byproducts and them not being properly removed during the subsequent purification steps. We also observed additional amino acids being inserted into the sequence for the GLP-1. So Endo-E is proteomic nomenclature to indicate that an additional glutamate was added into the sequence. And we identified aggregates of the GLP-1 agonist here as a dimer. For each of these, for the sake of time, I didn't go through each one individually, but for all of these, we were able to determine the sequence as well as the exact site of the modification that we say here. So when we say that there was an additional glutamate added in, we know where in the sequence that glutamate was added in. And if there is a modification on any of these amino acids, we know the exact amino acid in that sequence that was modified. So this information can be provided to the special agents or the investigators who are submitting these samples. Um, we can provide this immunity profile to them as a means to determine the source of potential counterfeits. Some of these impurities can also potentially elicit immunogenic responses in the patients, so they can have adverse health effects. And it also aids us in determining whether the products that are sent to us are authentic or counterfeit. So we will move on now from the analysis of peptides to how we analyze proteins. And you'll see most of what we do is going to be very similar, but there are some slight differences for how we go about tackling the larger proteins. So as an example, we're going to use human serum albumin, or HSA. This is a protein that we analyze a lot here at the NFCC because it's present in numerous biotherapeutics and vaccines, where it's added into the formulation to stabilize the active biotherapeutic. It can also be used as a delivery vehicle to carry the biotherapeutic to its intended target. And it's also used to protect the active biotherapeutic from degradation during the manufacturing and storage process. HSA consists of 585 amino acids, and it has numerous potential modifications, so it can have numerous potential proteoforms. So for proteins, what we want to know is the same as what we wanted to know for the peptides. We want to know what is the unique sequence of the amino acids of what we would observe in our samples, and does that match HSA? And we want to know what proteoforms are present. So are there any modifications to the HSA that we may observe in the sample? So as an example, we got in a vial of liquid that was labeled to contain a biotherapeutic protein as the active ingredient, and it was also labeled to contain HSA as an excipient. We were able to confirm the presence of the biotherapeutic using immunoassay techniques here in the lab, and then we sought to confirm the presence of HSA to determine the authenticity of the product. So we don't directly analyze proteins the same way that we do peptides. And the reason is a couple of reasons. First of all, it's difficult to produce fragmentation between all of the amino acids in a protein. So as these amino acid sequences increase in length, they will begin to fold over on themselves, and they form these sort of complex three-dimensional structures. And so it's difficult to find mass spectral parameters that will fragment at the amide bond between every single one of the amino acids. And if there's any region in this larger protein amino acid sequence that we don't have fragmentation between each of those amino acids, then that sequence in that region is ambiguous. We can't say for certain what that is. And the identity of all of these proteins and peptides is determined by their sequence. So if we can't determine the sequence, we can't say what the identity is. So so it's difficult to be able to induce fragmentation between all of them. Additionally, the MS2 spectra is much more difficult to interpret the larger the amino acid sequences are. So for a 15 amino acid peptide, there's only 28 theoretical B and Y ions that would be generated. Whereas for our HSA example, we would have nearly 1,200 B and Y ions that we have to account for in the fragmentation spectra. So what we do here in the lab, and what is most commonly done in the field of proteomics, is what's known as a bottom-up analysis, whereby we digest our proteins into predictable peptide fragments using an enzyme, and then we analyze each of those peptides in a single run the same way that we would analyze a single peptide. So in this case, we use trypsin for the analysis of our HSA, although there are other enzymes that can be used and that we do use here in the lab. But for HSA, we use trypsin, and it cleaves on the C-terminal side of wherever there is a lysine or an arginine amino acid in your sequence. So a theoretical protein sequence here, if it was reacted with trypsin, it would cleave at these two amino acids here, lysine and arginine, and you would generate 3 theoretical peptides. These peptides are then subjected to our LCMS analysis, and we would analyze each one the same as we had previously discussed. So here, our trypsin, our HSA is reacted with trypsin to produce our peptides. The peptides are analyzed via LCMS, and we look at the mass of our experimental peptides shown on the top, and we compare that with the mass of the theoretical peptides shown on the bottom. If any of these masses match, then we want to see, okay, does the sequence also match? And this is the same process we have done before, where we are just looking at the experimentally observed fragment ions from our peptide and comparing that with the theoretical fragment ions that we would predict from that peptide. If they don't match, then we say that it is not assigned to that sequence, even though the intact masses may match. But here we can see that our fragment ions match up with our theoretical ions. So we are able to assign this amino acid sequence to this peptide. And we can then map that amino acid sequence back to the larger sequence for our human serum albumin. So in this case, we were able to say the amino acids highlighted in yellow here are able to be accounted for in our analysis for HSA. We do this for all of the observed peptides, and in this case, we are able to obtain over 99% sequence coverage of HSA in the biotherapeutic formulation. And additionally, we only observed a single proteoform of HSA. We did not observe any modified forms. And these results were confirmed through analysis of a certified reference standard. But we don't stop there. What we want to do next is look to see, is there anything else in the sample that we might be missing? So far what we have done is a very targeted approach. They said HSA was in there based on the label, so we looked for HSA. But now we want to know, is there anything else present? So we want to do not only a targeted analysis, but we also want to perform an untargeted analysis to make sure that we don't miss anything. And if you go back and look at our raw data, if you look at the sample on the top, we see a number of chromatographic peaks in our sample that aren't present in our HSA standard. So we do see a number of peptides that aren't mapped back to HSA. So these peptides are coming from somewhere. So what else is in our sample that's generating these peptides? In order to ascertain this, we will subject the peptides that are not mapped back to HSA to an untargeted larger protein database. We have in-house databases that we use where we have all the known biotherapeutics, but we also will routinely search different publicly available protein databases. The most commonly used in the field would be the UniProt library, which has the amino acid sequence for approximately 600,000 proteins. And in this case, what we do is if we have our peptides that weren't mapped to HSA, shown on the bottom here, peptide 1, 2, and 3, and we subject this to an untargeted search across a large protein database, what we can see is the first two peptides, those amino acid sequences are observed in multiple proteins in the database. So while they aid us in determining the sequence coverage, we don't really have any discriminatory power because based on just those two peptides there, we can't distinguish which of the proteins they're coming from. So we can't say which of the proteins are in our sample. But if we look at peptide 3, we can see that that amino acid sequence is present only in protein number 1 here and not in any of the tens of thousands of other protein sequences that are in the database. So when we're performing an untargeted analysis, the unique peptides are really what we want. We are looking for getting hits for proteins that we are able to map multiple unique peptides to, because that gives us strong evidence for the identity of that protein in our sample and allows us to go and obtain a reference standard to run and confirm the identity of that protein. So in this case, with the sample that we had, all the peptides that we observed that weren't mapped back to HSA, we performed a large database search against the UniProt database, and we obtained numerous hits from multiple collagen proteins. Just as an example, one of the collagen proteins we got a hit for was the type 3 alpha 1 chain. 53 of the peptides in our sample were mapped back to this collagen protein, and 33 of those peptides were unique only to collagen. So what that means is 33 of our peptides had an amino acid sequence that is only found in this one collagen protein, and it's not observed in any of the other 600,000 proteins in the database. So this gives us very strong evidence that this sample has been adulterated or contaminated with collagen proteins. And I will just note here, many of these samples that we get are ongoing criminal cases with the Office of Criminal Investigations. So I am being somewhat ambiguous with what the biotherapeutics are. However, I can state that there is no FDA-approved formulation for this particular biotherapeutic that allows collagen proteins to be present. So this is indication that it's been adulterated or contaminated. So as the final example, we'll go through and see how we analyze glycoproteins and monoclonal antibodies. In the lab, we obtained had a sample submitted to us that was a pre-filled syringe that was labeled to contain a glycosylated biotherapeutic. So what these are proteins that have glycans attached to them at very specific amino acid locations. These glycans are large carbohydrate chains consisting of monosaccharides linked together. This particular biotherapeutic exists as a homodimer, where each one of the monomers are linked together to form a dimer via 2 disulfide bonds, and that is shown at the top of the figure here near the hinge region. And there is a single site of glycosylation on each one of the monomers shown on that red circle. And the glycosylation occurs at only this single site on this very specific asparagine amino acid for the biotherapeutic. So for the sample that was submitted to us, if we want to confirm the identity and presence of this biotherapeutic in the sample, we have to know not only what is the unique amino acid sequence and any proteoforms that are present, just like we've done with the other proteins and peptides, but now we have to know, are there any glycans present? And if there are any glycans present, what amino acid or amino acids are they attached to? So using our bottom-up proteomics approach, we were able to determine or account for greater than 99% sequence coverage. So we were able to determine the unique amino acid sequence of the suspect and confirm that that did match what was known to be for the biotherapeutic. And next, we need to determine if there were any glycans present. The way that we do this is we go through all of our peptide data and we try to identify if there are any peptides that have fragmentation mass spectra that show the evidence for these neutral losses of these monosaccharides, indicating that there is a glycan present on them. So in this case, we only observed one peptide that's shown in the upper left portion of that lower spectra there. Only one peptide that showed evidence of having any glycosylation, and we were able to localize that site of glycosylation on this specific asparagine amino acid, which is denoted in the blue letter N there. We noticed this mass difference on here, and we were able to elucidate the structure of the glycan that was on that asparagine by looking at the the monosaccharides that were fragmenting during the fragmentation process. So in this spectra, we are showing different B and Y ions. And the B and Y ions that carry that asparagine amino acid, we are also showing the structure of the glycan as it is being fragmented. And we can build this back up to show in the upper right corner of that spectra the structure of the glycan that is on that amino acid. Continuing this process, we were able to identify 9 unique glycans, so elucidate the structure of all 9 of them. And all of these were mapped back to a single amino acid, asparagine, at a very specific location in the amino acid sequence. And what we observed on the sample, our suspect, is also where it should have occurred on the biotherapeutic. The bottom graph shows a histogram where we compared our suspect sample with an authentic obtained directly from the drug manufacturer. And we see not only very good agreement between the structure of the glycans that we identified in our suspect and the authentic, but also on their fractional percent abundance matches very well between the authentic and the suspect. So based on this information, we are able to confirm the presence of this biotherapeutic in the suspect sample. But if we look at a mirror plot here that shows each one of the peptides that we were able to observe in our sample and our authentic, each one of these peptides, we see their masses and their corresponding intensity shown as a mirror plot here. And we see very good agreement between the sample and authentic. So as a just a cursory overview, you would say, yeah, they match very well. But if we zoom in on this region here, we actually observe a peptide that's showing up in our sample that we don't see in the authentic. So what is this peptide and where is it coming from? Well, looking at the MS2 data or the fragmentations data, we're able to determine the unique amino acid sequence of that peptide. It doesn't match our biotherapeutic, so it's not coming from the biotherapeutic, or at least we don't think it is. So what protein is it coming from? When we subject it to a search of the large protein database of approximately 600,000 proteins, that sequence is not observed in any of the amino acid sequences for any of those known proteins. So what we do then is say, okay, we know that no protein has this exact sequence in it, but instead we're going to perform a sequence similarity search. So we perform a search that says it may not be exact, but show us what protein has this sequence in it that most closely matches our suspect peptide. So it is allowing for one or more of the amino acids to be different. And so when we look at the sequence of our suspect peptide, the unknown, the hit that we get back says it's most similar to your biotherapeutic that's supposed to be in our sample. And the difference is the valine has been substituted to a threonine amino acid. Both valine and threonine have different properties, whereas valine is hydrophobic, threonine is a hydrophilic. This can cause changes in the structure of the biotherapeutic and can have effects on the function. So if we look at going back to our mirror plot, we can see that the natural form of this amino acid sequence is observed in the authentic and the sample, but this single amino acid substitution is only observed in the suspect. And in fact, when we go through and look at how much of our biotherapeutic has this substitution, we can see that for the authentic, none of it has the substitution in there. But for our suspect, approximately 40% of the biotherapeutic has this valine to threonine substitution at that very unique location or at that very specific location. This can have effects on the structure of the biotherapeutic, so that could cause a reduction in the efficacy of the drug. Um, Depending on the structural changes that could take place, this could elicit an immunogenic response in the patient, have an adverse health effect. And we can supply this information to our special agents and the investigators that we work with to allow them to begin trying to source where these potential counterfeit drugs are coming from. So today we kind of went over some of the proteins and peptides that we analyze here at the NFCC using mass spectral techniques. We went over peptides, proteins, monoclonal antibodies, and discussed some of the proteomic analyses that we perform on those. I will give the caveat that this is a very small snapshot of some of the forensic proteomic work that we do here at the NFCC. I have focused only on mass spectral techniques, and even then, only touched on some of the mass spectral techniques that we use. We have other mass spectrometry systems that we use and techniques that we will utilize. But for the sake of time, we just presented a small snapshot of what it is that we do. We've analyzed over 50 different biotherapeutic peptides, being able to characterize and elucidate the structure of over 20 different non-natural amino acids and different synthetic modifications. As you can imagine, recently we've been getting in a lot of GLP-1 agonists. We've been able to determine the unique sequence of over 7 different active pharmaceutical ingredients for GLP-1 agonists, 5 of which are FDA approved. We've analyzed 2 of them that are not FDA approved GLP-1 drugs. And we've also been able to determine the impurities in there and generate impurity profiles for these GLP-1 agonists. We've analyzed proteins, not only HSA, but insulin, human growth hormones, various toxins, ricin, and also monoclonal antibodies, drug antibody conjugates, as well as biosimilars. So as far as the future work that we do here at the NFCC for the proteomics, we are always looking to expand our database of any non-natural amino acids, known modifications for our peptides and proteins, and also doing impurity profilings and generating impurity profiles for all of the observed peptides and proteins for these samples that we get in to be able to provide that information to our special agents to allow them to source these counterfeits. Recently, we've been working on bacterial proteotyping, which is utilizing LC-MS to rapidly identify and characterize bacteria in samples. However, what we are working on is really dictated by the samples that we get in. So I will say this is currently some of the work that we are doing, but if next week we get in a bunch of monoclonal antibodies, then we pivot and now we're doing monoclonal antibody method development. So what we are doing here at the NFCC is dictated by the samples that we get in and what is needed from us. So with that, I will thank the analysts here at the NFCC who assist me and do the lion's share of the work. That's Robert Chapman, Rachel Dickin, and Samuel Seibert. I wouldn't be able to have done this presentation without their help in the work that we do each day here together. Thank you to our management here at the NFCC. And I'll just end it by saying, on behalf of the National Forensic Chemistry Center, I thank you for allowing us to talk on some of the proteomic work that we do here each day.